Thanks for everybody for listening in on this really exciting area of spatial biology. So let me just describe to you how we invented and how we developed this GeoMx technology. And literally, a lot of us just came from American Association for Cancer Research meeting in Atlanta just from a couple of days ago. And it literally was 5 years ago, almost to the date, 2014, where me and a professor from MD Anderson, a guy named Gordon Mills, we scratched out on a napkin essentially what are the essential features of the digital spatial profiler. And I'm trying to highlight them here on this particular slide. The key aspect that the problem we were trying to solve was, my background is in next generation sequencing, so I'm used to getting 10,000 answers at a time or some big number. And then I sit here and I watch people like Sergio come to me with these absolutely gorgeous, precious tissue slices. I put them in a microscope and I get 4 answers or 8 answers or 10 answers. I wanted 10,000 or I wanted 100,000 answers out of those things. So what we needed to have was a microscope that would allow you to have unlimited plex. To be translationally relevant, it had to work on FFPE tissue. And to get all the information we wanted, it had to work for both RNA and for proteins. So we came up with this design strategy. If you look on the left-hand side of the screen, all the other technologies, or many other technologies, were trying to stuff the assay into the tissue. And we said, we're never going to get 100,000 dyes to actually give me an answer in a piece of tissue. So what we did is came up with this idea that you would attach a small oligonucleotide sequence to your affinity reagent. You can see either an antibody on the left-hand side or a FISH mRNA probe. And then the key trick, if there was, on the DSP is that we're going to connect those little indices with a photocleavable linker to the affinity reagent. What that allows you to do is you can actually go ahead and saturate a single FFP slide in one single step with as many independent antibodies or as many independent FISH probes as you wish. All those probes are invisible, so you don't really see anything in the microscope. So what we do is we take those thousands of probes, we go onto the tissue slice, and we dope in, say, 4 fluorescent colors that allow you to see the overall tissue geometry, like you can see on the right side of this slide. And then the microscope can take those pictures in 4 colors and look at interesting aspects of the tumor or the tissue you're looking at, and then move the microscope to that region where you want to do your high-plex profiling. You turn off all the fluorescent colors, and you shine a brief burst of the UV light. The UV light then in these localized regions, you can see those little circles on the tumor slice there, will then locally release all the FISH probes that are in that particular geometric aspects of the tumor, and the microscope literally drinks them right off the surface of the tissue. So in that way, we moved the assay-dependent part of the signal to just these small oligonucleotide tags, and then they are read out, as you'll see here, either using the normal N-counter instrumentation up to 800 plex, or when you want to go to unlimited plex, you read out those probes on a next-gen sequencer. So let me just take you very quickly through the overall flow you see on this slide. Starting at 1, you put in your high-plex cocktail of FISH probes or antibody probes. They're invisible. You dope in some fluorescent colors. That gets you into step 2, where you can look at the overall geometry of the tissue. You find your interesting regions of interest, step 3, and you shine on the 385 nanometer light. That releases all the photocleavable barcodes associated with those affinity reagents at that localized geometry, and you literally drink them off the surface of the tissue at step 4, and you deposit them onto an on-instrument microtiter plate. That entire process from step 3, 4, 5, and 6 repeats and can be accomplished in about 30 to 45 seconds. What this allows you to do is you can just move through the geometry of the tissue, generating what I call high-plex digital replicas of the affinity reagents in any of those locations. So that's the overall essence of how the GeoMx system works. Now, one of the unique aspects of this particular microscope is that it profiles, it actually will record an image, like you see in the center of this particular slide, which is a colorectal tumor tissue microarray. I take a picture of that, and I can dope in the fluorescent color markers, but they can be doped in a very unique manner. So this is pan-cytokeratin you're looking at. So the yellow signal will light up all the tumor-specific cells. Now I wanted to be able to profile automatically just those tumor-specific cells in a pure manner. So in the back of the GeoMx instrument is adaptive optic. It's actually 2 small digital micromirror semiconductor chips with about 1 million mirrors. That allows the microscope to actually take a picture. That picture can actually be converted into a mask. And so if I'm taking a picture of pan-sided carrot, you can see in the upper right-hand side, that represents the pure tumor signal. That pure tumor signal, actually that image directly drives the semiconductor mirrors in the back plane of the microscope. And they will immediately automatically reorient themselves so that the molecular profiling light only touches the tumor-specific tissue. So in one fell 30-second operation, I will get a pure tumor signal of the antibodies or the FISH probes that are on only the tumor cells. Now the beauty is that you can do mathematics on these masks. So if I invert that mask and then repeat that process, what will I get? With single cell resolution, I get pure antibody profiles or pure mRNA profiles of only the tumor microenvironment. So 2 30-second operations, and the microtiter plate is the antibody profile of the tumor only, and the compartment, the microtiter plate, right next to it would be the pure tumor microenvironment. So that's one of the key features of the microscope as it gets used in immuno-oncology. But obviously there's ramifications. If you're a neurodegenerative disease, I could be actually imaging, say, Alzheimer's plaque, and then it'll automatically configure itself and you will be profiling, say, Alzheimer's plaque, just like you would a tumor. This gives you an idea, and you'll see some more of this images especially from Aubrey. But the idea here is that the instrument will self-configure. So this is an excisional biopsy. I take that picture, and then doing something like Sergio was talking about, I'll light up the tumor-specific cells. This is all done automatically, and then the mirrors will reconfigure and shine light on just the green shaded region to give you a pure tumor signal. At the same time, it'll auto-configure. The mirrors will change configuration and give you a pure signal out of the tumor microenvironment, all in an completely automated fashion. Now, the nice thing about having an adaptive optic in the back of the GeoMx, when you're talking about flexibility, every time we put a tissue slice in there, it can self-configure itself over all these different types of geometries. We allowed the pathologists to actually sit there and scribble right on the tumor themselves and do geometric profiling, automated segmentation where the instrument automatically reconfigures itself. You can do rare cell profiling. Then you do some very unique things where the microscope will automatically configure itself around particular boundaries in the tumor, or you can just do gridded spatial imaging. But all of these things are auto-configured, and the microscope absolutely adapts to how you want to interact with the tissue and will configure themselves in all these different automated segmentation modes. So like Nero was telling you, we've been running this. I hand-built these 3 years ago, and then we would have these visitors come in, and literally the pharmaceutical companies wanted to buy those prototypes right out of my lab. I could not sell them that. So what we did is we allowed them to send us samples, FFP samples, through the mail, and then we've been running them for a couple of years now. And you can see all the different types of cancers that we've studied and the distribution between academics and pharma. We learned a lot from doing those programs, and they really helped us build a better instrument. I think probably David Ram our instrument because it taught us how pure we had to do the compartmentalization, and it actually was the reason why we put 2 semiconductor mirrors in the microscope instead of 1. The other thing we worked very hard on, and this was like listening to people like Aubrey and Sergio, was that, the microscope, we wanted this microscope to actually represent the biology, the final biology you're trying to do, which is how do you generate biomarkers that predict response? The microscope's got to know how to do a Kaplan-Meier curve. It's got to be able to import that kind of metadata. So we incorporated all those kinds of features in. The other key aspect is that we wanted to make sure that when you do your molecular profiling, we wanted to always maintain that connection between the molecular profiling and your raw data. So that when you see a volcano plot, and you'll see a bunch of them, you'll see some great ones from Aubrey Thompson for triple-negative breast cancer, where you actually can circle your key biomarkers. And when you do that, it'll automatically take you back to the raw images that generated that data. So you can always go from profiling into the tumor again and back and forth. This was a key aspect of the way we designed the software for the system. Now, this is a reasonably mature system now. I've been talking to you, especially from the protein side. Our very first 2 papers on the DSP actually were both back-to-back in Nature Medicine, October 2018. And actually, it was really nice. The very first DSP paper had the Nobel Prize winner, Jim Allison, in immuno-oncology on our very first DSP paper. Genentech actually wrote a review on this technology before we even launched it. It's in the Journal of Pathology, which is very unusual. I don't think that's ever happened before, to have a review article before you even launch the technology. Now, I'm going to have to go really fast here because Nero took some of my time, but I will try to get it back. I'm going to show you a little bit more on where the instrument is going in the future. And this is the near-term future. When the microscope was developed, you can see we actually developed it so that was a multi-analyte microscope. You take 1 5-micron slice and you can do your high-plex protein profiling, like I described here. And that protein profiling goes, to give you an idea, typically our IO detection modules are about 100 or 150 antibodies on a particular kind of biology type. And then the microscope is set up so it takes an adjacent 5-micron slice and allows you to do high-plex RNA profiling. The microscope will actually orient those images with single-cell resolution so the geometry is maintained. And then in the analysis, you got a totally combined analysis with the biomarkers from the RNA layer, the protein layer, and the protein post-translational modification layers all can be analyzed in a single integrated step. And this was really the heart of what we're trying to create. One thing that surprised us about RNA, and Omar really brought this up, is the scalability of the multiplex in RNA. I remember when I first started doing these experiments, and I went to my scientists and said, we're going to do 100 RNAs at a time. And they said, you're crazy? You can't do 100 RNAs at a time. We did the experiment, and it worked. And I said, okay, well, then I'm not happy with 100. Let's do 1,000. So we did 1,000, and when I did 1,000-plex, I embedded the 100-plex inside of it, and I looked at the correlation. These correlations were all greater than 90%. So we just kept going. So we didn't stop at 1,000. You can see that by the end of this year, I'll show you some data actually today on 5,000 independent RNA tags all resolved simultaneous in space. And then this represents about 1,500 genes. And we are now exploring taking the technology all the way to full transcriptome. We're going to look and probe the ability to do 24,000 genes at a time in space with RNA. That's the real power of RNA. Let me show you what it looks like. When you do this kind of work, then we're going to say, how do you represent 5,000 independent RNA probes in space? One of the ways we did this was actually, what you're looking at on the screen now is the entire reactome. It sort of represents cell and tissue biology on one screen in terms of all the pathways that you could probe. Now, if you look at when you're doing 5,000 independent RNA probes, you can see all the individual pathways that we're covering in this reactome. It's really a very nice dashboard for what the tissue is doing. So then we actually get to the point where this is when me and Gordon scribbled this thing out 5 years ago on a napkin. This is where I wanted to be. I called it pathways in space, and now we're really measuring it. So you can actually see this is an excisional biopsy, colorectal cancer here, and I'm actually measuring, you can see 5,000 RNA probes spatially resolved. The pathologist looking at it actually denoted the invasive margin, and then the pathologist at the low plex, they looked identical. But then when you actually look at the reactome, look at the bottom graphs there, you see the one invasive margin to the right there, the immune signaling and the whole cell signaling part is totally dead. It's actually way low in mRNA, whereas the invasive margin right next to it, you see in the lower left-hand side of the screen, is totally active in both the immune signaling and signal transduction. So this gives you an idea. It's a window into pathways in space that allow you to see these kinds of effects. And then when I was looking at why is that one region invasive margin hot and the other one is cold, all I did was dial up all the chemokines and cytokine mRNAs that I have in there. And this gives you an idea. This is a little window into the chemokine environment of those 2 particular regions of that colorectal tumor. Now, it was very interesting as I just got back from AACR, and I had a person that I was actually showing this raw data to, a guy named Jerome Galon, who sort of invented this whole spatially resolved IO assay. It was called the Immunoscore Consortium. And he looked at that data and he said, Beechem, I know that. That's a result we found out in 2016, and we published it in Immunity, and it had to do with the anomalous behavior of CXCL14 and 16. So I said, that's interesting, Jerome. So he immediately looked. So what I did is I actually looked into Jerome's original publication. There it is, the Immunity in 2016. And look at those anomalous. It's actually a very interesting effect that happens in these tumors, these MSI high and low tumors. And I don't have time to go into the biology. But normally the MS, the microsatellite unstable ones, are all hyped up in the chemokines and cytokines. But one of them runs backwards. And the one that runs backwards is the CXCL14. And Jerome was so worried about that, he saw it in RNA-seq, and then he had to validate in qPCR to tell him it was real. And then when he looked up on the screen, that's exactly what shows up in the DSP. The CXCL14 all runs backwards, and it's the same exact thing that Jerome was able to find in his original study. So it actually gave me a lot of, when you see independent scientific discoveries like this, it really gives you confidence that the technology you're measuring actually it's working as designed. So I think I'm going to end it just about here. It's sort of a summary of the overall aspects of the instrument. There's multiplex that does many analytes on one tissue, and you can see with the next-gen sequencing readout, it's infinite. It's infinite. I use 12 base pairs to do the indexing. 12 base pairs is a little over 16 million things you could multiplex at a time. So that's pretty good compared to what we used to do in I8C. This guy goes to 16 million. That's multi-analyte. Both protein, RNA, and protein post-translational modifications can be done. And you can see PTMs inside FFPE tissues. It's quantitative. I didn't talk too much about it. This is a true digital chemistry. So it's not an analog chemistry. So the numbers you get out here have a huge dynamic range, and it's all digital. The limits of detection for protein, certainly you go right down to the single cell limit. The hardware actually works to about 1/10 of a small T cell. 1 square micron is sort of the spatial resolution of the hardware. It's non-destructive, so the sample is only touched by light. So when I'm done, I can pull the thing out, store it wet, and then you can come back and reprobe it. And then this was a key thing, because we really listened to our clinical customers, is that you got to be able to have some high throughput here. You know, I can't spend a half a year looking at a clinical trial cohort of a couple hundred patients. I got to get that data in a week and a half, 2 weeks, and be done with it. So we designed this instrument to run at roughly 20 sections per day. So this is my very final slide. Obviously, NanoString's a relatively small shop. We got 200 people in R&D. There's about 500 people in the company. So we actually partnered with a variety of groups that will help us to really scale this biology. We announced all 3 of these at the AACR. So Beechem is now going to be actually making these GeoMx branded antibodies that will actually work in the GeoMx. So it immediately gives you access to 6,000 I8C qualified antibodies for you to actually add to your panels. We actually work very close with high throughput automated process. So the Leica bond system, there'll actually be a little menu item that says GeoMx something RNA and GeoMx protein that will actually do the entire automated processing for the GeoMx. And then finally, we partner with ACD, who developed this technology called RNAscope, which is a great technology marker for our ultra high-plex RNA. So you can visualize with an ACT 4, 5, 6, 7, whatever panel, and then superpose on top of that 20,000 RNA probes if you wish. So anyway, that's an introduction to the technology. I'm a little bit over, but anyway.